Can you hear me? Okay. Well, I want to thank Michelle and the Institute for inviting me. This has been a great pleasure to come here, and I really enjoyed the morning sessions and this afternoon. Michelle asked me to talk about a relatively new area in the bone field. I'm using a different one. Yeah, this. No, that doesn't work. No, that's got my computer. Well, yeah, continue. Anyway, as I said, this is a relatively new area. As bone biologists, we've spent probably the last 50, 60 years studying the role of bone and calcium and phosphate metabolism, but studies over the last 15 years have indicated that bone plays also a role in bioenergetics and overall energy homeostasis. And this really came out of kind of accidental observations in genetic mouse models in humans where bone proteins were manipulated and fat resulted. And so, in fact, that's how we got into it about 10 years ago when we made a mouse, and I'll show you how our own experience and how we actually stumbled into this field. Oops. So I really want to cover three general points. First, as I said, I'll show you how we made this accidental discovery on the role of insulin-induced osteocalcin, a bone protein, and how it regulates pancreatic secretion of insulin and therefore overall energy homeostasis. And after we made that discovery, we became kind of asked ourselves, well, why should bone care about what's going on in peripheral metabolism? And so the second part I'll talk about studies aimed at asking questions about what types of fuel bone cells use and how much they need, and why does it make a difference. And then in the third part, I'll just show you a brief example of how at certain stages in the bone cell's life cycle, it actually makes citrate from the TCA cycle, and that's used for incorporation into bone and its structural integrity. Now, I'm going to show you a lot of mouse experiments. I'm a mouse doctor. People talked about being mouse or human, but don't dwell on the details or the mechanisms. The technical parts are not the point, but I threw them up there in case anybody's interested. The point is the models that we'll talk about, and I'll try to make that clear as we go along. So I thought, though, it would be first useful again to orient ourselves with bone cells and not chondrocytes, which we heard earlier about, which populate the growth plate. We'll talk here about the cells of bone, and they really are three types. The osteoclast, which is this multinucleated cell derived from a monomacrophage lineage, comes from the circulation, and that's the cell that resorbs bone. Then the cell that we'll talk about most today is the osteoblast. These are mesenchymal stromal cells related to cells that become adipocytes and muscle cells. And these are the cells that form bone. And then there's a third cell. The most populous cell in bone is a terminally differentiated osteoblast that actually becomes embedded in its own mineralized bone and is thought to be a sensory cell, sensing nutrients, sensing mechanical loads, and so forth. But the fact is, we still know very little about what this cell does because they're very difficult to study. Once you get them out of bone, they die. So they've not been very available for study. But I'll be talking, all the mouse models and genetic manipulations I'll show you today are made in this cell and the osteocyte. So we're studying basically the osteoblast, and I won't talk more about the osteoclast today. Not that it's not important. So I thought, just again, to reorient you to with what bone looks like and with the consequences of bone. So as we age, and you're not seeing the disappearance of the trabecular bone, I don't know why the movie didn't work, but we make all the bone we really have in early adulthood. 20 I put up there, it could be 20 to 30 years, you make all the bone you're ever going to get. And you then lose it, and men and women lose bone both lose bone. Women lose it more rapidly over the menopause. We all end up with a lot less. And once you get down to these levels, you start fracturing because you just don't have the strength anymore. Most of the structural strength is in your trabecular, what's called spongy bone, in the vertebrae and in part of your hip and so forth. That's in the center of the bone, and that's what's really lost. There, I guess there it goes. Now, I'm supposed to go together. So that's kind of bad news when you start losing that, because at that stage you're susceptible to fracture. This is what it looks like in a cartoon that's animated, and I guess I can't. Can you start that for me? Maybe if I press this. Oops. There you go. So you'll see precursors of osteoclasts coming from the circulation, and they'll settle down on a bone surface that needs repair. Bone is remodeled constantly to repair damaged bone, and you basically turn over your entire skeleton in 10 years. So here are the osteoclasts excavating a pit, and this occurs stochastically throughout the skeleton. The osteoblasts, shown in blue, come along and fill that pit. And so that's how bone is constantly remodeled. And if everything goes well, you put that packet back and it's fine. But the fact is, you lose bone because the osteoblast, first of all, the osteoclasts dig bigger holes, and the osteoblasts can't fill them. So you end up with a net loss of bone. And that's the problem with structural changes that occur. By the way, all the drugs that have been designed to treat osteoporosis are based on the knowledge of this remodeling cycle, mainly anti-resorptives to stop the osteoclast, but now new anabolics that work on the osteoblast as well. So let's talk about the first kind of serendipitous finding that osteocalcin is an endocrine factor made by bone. And again, we were interested for a long time in trying to distinguish actions of two related growth factors, IGF-1 or insulin-like growth factor and insulin. And these are very evolutionarily conserved molecules and receptors. In fact, in worms and flies, they're a single receptor with many ligands. But as we diverged, they became two separate receptors and are served by different ligands, insulin and IGF-1. And we tend to think of insulin as an energy regulating molecule, and IGF-1 is more of a growth and differentiation molecule, but that is very simplistic, and the fact is there's still a lot of overlap in the pathway. So what we wanted to do is to make models that lacked one or the other receptor, and that way we could study the effects specifically, or at least the effects of loss of each signaling pathway in a mouse, in a real mouse. And we did that by making mice, and again, don't get into the details, they're not important, but mice that either lack the IGF-1 receptor only in the osteoblast, so not in any other cell in the body, in the osteoblast alone, and the same technique was used to make one that lacked the insulin receptor. So what was the effect on bone and other sites of that genetic manipulation? Here you can see micro-CT images of a control, an IGF-1 null and an insulin null. And if you look at CT images really at 6 weeks, they don't look much different. But if you do quantitative histology, there's a big difference. The IGF-1 knockouts have actually more osteoblasts. I won't get into the reason, it's probably compensation. The real problem there is a mineralization defect. They can't mineralize the bone that they make. Again, I'm not going to talk about that right now, but it's an interesting observation which we're actually studying. Notice the big difference here, the mice that lack insulin receptors in their osteoblasts lose 50% of their osteoblasts, and that leads to this bone loss. They don't have any problem with mineralization, so a very different, dramatic phenotypes in both animals, but strikingly different, suggesting that these molecules both work in different ways. The real surprise came when the graduate student who was working on this went down to the animal room and he noticed that the mouse that lacked the insulin receptor was getting fat. And I didn't believe him. I told him to go check again and again and again. He was right. That never happened to the animal that lacked the IGF-1 receptor, despite its homology. And you can see here by QMR that these mice gain a lot of weight starting at around 8 to 10 weeks of age. It turns out that they become hyperglycemic and hypoinsulinemic. And again, this is like somebody who has basically development of peripheral diabetes or insulin resistance, although this low insulins, we have a reason for, which again, I'm not going to get into, but this is a metabolic defect, and we went on to formally test their glucose tolerance using glucose tolerance tests, and the mice that lacked the insulin receptor became glucose intolerant, and they were very insulin insensitive compared to controls. So that is extremely interesting. Remember, the insulin receptor is lost only in the osteoblast. Everywhere else it's normal. So this mouse had two strikingly different phenotypes than the other mouse I talked about, a bone defect due to low numbers of osteoblasts, And then this development, what looks like when they became fat, peripheral adiposity and insulin resistance in the periphery. So what in the world was going on? So what we did is we used a classic technique which everybody uses now. This was an old way of doing it. We did basically a microarray, or profile the genes in the cells that actually either lost the IGF-1, were treated with insulin or treated with IGF-1. And of the 470 genes that were insulin responsive by 2-fold up or down, the one that really grabbed our attention was this protein, this gene here called BGLAP, and that basically is osteocalcin. That encodes a gene called osteocalcin. Osteocalcin, so I should say at this stage, we were scratching our heads. We had no idea what we were looking at. We sat on it for about a year, and then a paper came out by Gerard Crescenti's group showing basically that osteocalcin knockout mice had exactly the same phenotype we had in our insulin receptor knockout mouse. And to make a long story short, it ended up that the osteocalcin was a secretagogue for insulin. So we had the opposite end of the pathway. Basically, osteocalcin is a protein that's made only in the osteoblast. As I showed, it was stimulated by insulin. And normally it is involved, it's thought to be involved in the mineralization of bone, and it stays, because of its high avidity for bone, it stays close to bone. It doesn't escape the circulation. But there's a post-translational mechanism whereby it can lose that affinity for bone, and some of it escapes into the blood, and that's what's called the endocrine form, or undercarboxylated form of osteocalcin. And so this is what Dr. Crescenti had actually hypothesized, that this endocrine form was the hormonal form that was interacting with the pancreas and basically promoting insulin secretion. So to link the two groups of work, what we had to do is show that this undercarboxylated form was lower in our mice, and indeed it was. These are serum total osteocalcins in the knockout mouse that lack the insulin receptor, and these are the undercarboxylated levels. And then what we did is we came back and administered this undercarboxylated, quote, hormonal form of osteocalcin to the mouse that lacked the insulin receptor, and it improves the actual, in this case, the insulin sensitivity in that mouse. So again, this strongly suggested that this osteocalcin was indeed made by bone, responsive to insulin, and was acting in the pancreas to promote insulin secretion. And there's been a lot of studies by Gerard's group, a number of other groups, to show that this really is acting, at least in mice, as a hormone. So it acts, as I said, on the pancreas to promote insulin secretion. It acts on brain through a different receptor. It acts on fat, muscle, and on the testes, at least in male mice, to promote reproduction. So this is quite an interesting hormone that I think is going to have to be studied for many years to come in humans. I think that's the real critical thing, how much of the mouse work relates to humans. But it emphasizes what's a part of a larger story that's emerging, that bone cells, and particularly the osteoblast and those osteocytes that are resident in bone, probably act as glands and sense prevailing energy needs, and then make hormones just like other tissues do to sense inventory and fuel supply, and then communicate that information to other centers of control. So that brought us to a whole second series of questions, as I indicated, as to why should that matter? Why should bone be paying attention to global energy balance and fuel? And so we started to look at what fuel osteoblasts need and how it's regulated. And obviously the first thing we looked at was glucose, because that made the most sense. And we found that like other cells, like muscle and fat, insulin stimulates the uptake of glucose and its oxidation. These are osteoblasts in culture. Interestingly, insulin stimulated the uptake of fatty acids, but not its oxidation. I'll come back to that, because fatty acids are used by osteoblasts, but they're regulated through a different mechanism. So how much glucose does an osteoblast need compared to other tissues? What we did is we were able to use non-metabolizable FTG glucose and just look at distribution after an ejection. And you can see here these red bars indicate how much of the dose of FTG-18 labeled fluorinated glucose is taken up by bone tissues, and a relatively large amount, if you compare it to these other tissues, actually taken up by bone. If you knock out the insulin receptor, notice the red bars, you really reduce that uptake of glucose by bone. And you can even image using PET-CT where that FTG-18 fluorinated glucose ends up, and it's at the ends of the bones. It's really not in chondrocytes, although it looks like it might be in the growth plate. It's really in the metaphyseal area. In fact, that's the site in bone, in the long bones anyway, that the most of the osteoblasts are actually located in the mouse at this particular time. So it fits with this idea that osteoblasts at these sites are taking up a lot of glucose and assumingly using it. So we looked a little bit at the mechanisms, and we took our page out of the other work that had been done on the regulatory factors that how does insulin regulate glucose uptake. And we found that in osteoblasts express a glucose transporter, GLUT4, just like muscle cells do. When insulin binds its receptor, it promotes translocation of that glucose transporter to the membrane, and that's how glucose enters. So what we did, again, we took a genetic approach, and we said if it's important, if you knock it out, what happens to bone and what happens to the other, to fat and muscle and so forth. And so again, we made a mouse that again lacks this glucose transporter, and again, I emphasize, only in the osteoblast and no other site. And we were a little surprised that the early data so far indicate that this does not have a disastrous effect on bone. So it's not seemingly affecting bone density by micro-CT, but in fact, they gain weight too. So the mice develop peripheral adiposity. And in fact, if you do an insulin tolerance test, they become insulin resistant. And we did actually an insulinemic clamp, euglycemic insulinemic clamp on these mice, and we got evidence for the decreased disposal of glucose in these mice. It's a little different what I showed you before. These mice, probably because you've taken out the major source, the major site and mechanism of glucose uptake by the osteoblast, there's excess glucose, it has to go somewhere, it probably ends up in fat and the disposal goes down. Again, indicating that osteoblasts are, if you are a major user of glucose, if you can affect, at least if you believe the data, if you can affect overall glucose disposal simply by removing that transporter. Now, I mentioned lipids are also taken up and used by osteoblasts, and this is data from Ryan Riddle in our group. And here you can see that lipid oxidation increases if you put osteoblasts in culture. They differentiate, and as they differentiate, the oxidation of lipids goes up. You can inhibit that oxidation with an inhibitor of lipid oxidation, ETO. When you do that, you affect the differentiation profile of the osteoblasts. They don't fully differentiate. Just these are the control cells, and the more red you see is the more differentiated they are. So it knocks out the differentiation. And then again, you can use techniques to look at in vivo labeling, and again, fatty acids end up in bone compartments in vivo after injection. Not as much as glucose in this experiment, but still to a great extent relative to the other tissues. Now, I'm not going to get into, this slide really is again to show you, well, how much does fatty acid metabolism mean to this animal? How much do they use? And again, the details aren't important, but we were able to knock out a rate-limiting step in fatty acid metabolism and knocking out CPT2, an enzyme right here in the pathway. And that's really a rate-limiting step in the fatty acid metabolism. When you do that, you actually decrease bone in these mice. So again, suggesting that fatty acid oxidation is critical for osteoblasts to really lay down bone and make mineral. You remember that I showed you the glucose transporter 4 mice didn't have much of a bone phenotype, and one of the possibilities is that they can compensate through fatty acid oxidation and therefore use this type of fuel when they don't have enough glucose. Now, insulin doesn't really stimulate, as I mentioned, it doesn't stimulate the oxidation of glucose, so what's of fatty acids, so what does? And again, almost through a serendipitous finding, Ryan Riddle's group found that that oxidation of lipids by osteoblasts is controlled by the Wnt signaling pathway. I'm not going to get into the details, but the Wnt signaling pathway is a major pathway preserved from flies to us, and it's really responsible for the development of the osteoblast. In fact, there's a drug on the market indicating that the manipulation of this pathway can work as an anabolic agent. And when he actually studied a mouse that was lacking this receptor, the ability of the receptor to work because he knocked out one of its components, obviously he saw low bone mass. We kind of knew that, but these mice also became fat. Here are the fat pads. Again, a shift in body composition, presumably because you've knocked out a pathway. Well, he went on to show, and I'm not going to show you all the details, that this co-receptor for the Wnt signaling pathway, LRP5, is absolutely critical for taking up fatty acids in osteoblasts. If you don't have it, you can't burn fatty acids in osteoblasts. So this was really critical, and again, indicating the importance of fatty acids in bone formation. So to summarize these two parts of the talk, the first thing is that osteoblasts can oxidize both glucose and fatty acids in amounts that really impact whole body substrate availability, because I showed you if you can manipulate through genetic approaches either glucose uptake and oxidation or fatty acid uptake and oxidation, you then shift body composition as well. And so that's really, I think, the first time that that's really been demonstrated, and it really has implications, as we'll talk about in a minute. The second summary really is this, and that is there seems to be a coupling of developmental signals to the bioenergetic program. Let me explain that a little bit. And again, I'm not going to show you all the data we have, but I showed you the data for LRP5 and Wnt signaling. So as osteoblasts differentiate, here you have an early osteoblast whose main responsibility is to divide, okay? That needs some type of transcription factors, and they use glycolysis and take up glucose mainly, not our work. As they move to a cell that needs to make a collagen matrix and then mineralize it, and think how unusual that is for a cell in the body to make tons of collagen and then put down mineral. There's no other cell that does that. They switch their bioenergetic program, and one way to do it is using this developmental factor LRP5 through the Wnt pathway, and then they change their fuel preference to some extent and begin to burn fatty acids as well. Terminally differentiated osteoblasts or osteocytes, we have no idea what's going on. They're hard to study, although we'd like to get at that through techniques I won't have time to talk about. So again, the same developmental programs and genes that were responsible for the differentiation of osteoblasts are coupled to the bioenergetic program that's appropriate for that particular job that they have to do. And then just in the last 2 minutes or so, I just want to talk about a specialized bioenergetic program for bone mineralization, which we found really in the last year we've been working on this, and not just from our own work, but through studying the literature, even literature going back to the 50s, where people were interested in, more interested in the bone as a metabolic organ than they have been recently. And I'll just talk about it briefly. It turns out that, again, reading other people's older literature, reading the older literature, citrate is present in your body. Most of the citrate that's present, up to 80%, is in your bone. So bone is made of collagen, and then it's mineralized by adding hydroxyapatite interspersed between those collagen fibrils. Hydroxyapatite is calcium phosphate, but in that hydroxyapatite, it's been known for years that there are other small molecules. Zinc, citrate, so forth, are there. So NMR studies by a number of groups have shown that citrate is actually incorporated into that. And so we were intrigued. Why should there be so much citrate in bone, and what's it doing there? And so what we found talking to people who work on a prostate cell, where also this mechanism seems to occur, is that calcium can come from the blood, but citrate cannot come out of the blood to go into bone. And indeed, it's interesting, it has to be made through the TCA cycle. So one of the jobs of the TCA, if you'll remember your biochemistry that we all took when we were students, the TCA cycle, one of its jobs is to make more ATP, but in the process you get citrate. Normally that citrate goes around and keeps making ATP, but in bone and prostate cells, it's diverted out of the osteoblast or out of the prostate cell. So these cells produce citrate, and we believe bone actually does it because it needs to get into bone for strength. And I'll just, again, show you one example of how we've proved this, I think, at least in vitro. So again, don't have to dwell on the details, but they're a way of using C13 heavy isotope labeled glucose, and we use uniformly labeled glucose and then follow the C13. Where does it end up? And what we can do is we can show, in fact, it ends up basically in, this is, over here is, the light blue bars are actually the C13 citrate that is in the mineral under the osteoblast after we cultured the osteoblast for 14 days. So you can see that they accumulate over time citrate in the bone mineral itself, just as we proposed and just as was speculated 50 years ago. So I think this is really an exciting finding because for the first time it suggests that the bioenergetic program of the mineralizing osteoblast is highly specialized. And you don't take citrate out of the TCA cycle and compromise ATP without something else going back in, and we think probably that's basically compensated for by the interjection of a number of other intermediates during that pathway. Glutamine is one, for example. Aspartate is another. So bone is a citrate-producing cell, as is the prostate, but there are very few that are. So I'm going to just really kind of end and sum up, and then I'd like to spend a lot of time for discussion if we have it. I showed you that intermediary metabolism of the osteoblast seems to be programmed by developmental pathways, and that's not my idea. That's pretty well understood now in other tissues and in cancer, where the bioenergetic program really is coupled to the developmental signal. So like with Wnt, you get that effect on fatty acid metabolism, but it allows the cell to then adjust its bioenergetic and fuel uptake needs to what it's doing at the time. Does it need it for division of DNA? Does it need it for making mineral? And does it need it for survival and whatever the osteocyte does in terms of its sensory thing? That seems to be what's happening, and that makes sense. The other thing I should mention is bone. I think we haven't really realized this, but bone cells, if you count up all the numbers of osteoblasts as a mass, and you extrapolate to some extent and compare it to other tissues, we calculated that the total mass of osteoblasts in a skeleton is about 1/4 the size of muscle mass. That's a lot of cells. And given what they have to do at certain periods of the life, we think that really makes sense, that you really do need a fair amount of fuel, and therefore you're competing. Bone is always competing with other tissues for energy fuel. And the third thing is this because of this, we believe that that's why these endocrine pathways, which were really unknown until a decade ago probably, need to play a role also. So you not only need to adjust locally your fuel uptake and metabolism, but you need kind of endocrine control. Otherwise, how does the cell inventory what's on board and communicate that to other tissues so that they can adjust whole body energy expenditure to what it needs? And then finally, I think this brings up the kind of the fairy tale, if you want. If you think about therapeutics, there won't be anything going on for a while because there's some really good bone anabolic agents coming along, and I think drug companies are moving on to muscle and so forth, but they'll come back. And if they come back, it would be interesting to target two different things. You could conceivably come up with a drug that would make a thrifty phenotype and also have good bone. By knowing this information, you could actually come up with a molecule that should be able to do that. I think that's going to be the challenge in the next 10 years to find out what that might be, what are the signals and so forth. I should also mention one caveat in all of the things I've been talking about. We work with mice. The other challenge is to figure out how much of what I showed you here actually pertains to humans. I'm optimistic on several areas. One is that there's a lot of association studies that have really confirmed, at least so far, this osteocalcin insulin connection. Again, it's association studies. They have to be proved, but it's harder to do that. That's number one. And number two is, GWAS was talked about, this genome-wide association studies have found a great number of genes that come up in screens of adiposity, body composition changes, and they overlap with bone density. And we're working on a couple. So there seems to be even inherited loci that are actually encoding genes that control both of those. So I still think there's a caveat. The mouse is a very highly metabolic critter. I mean, it eats 1/4 of its body weight every day to keep warm. We don't do that. So that's one caveat, that probably the mouse is very sensitive to these changes. But I think some of this is going to turn out to be important in humans, and that's what we're going to be working on in the next 10 years. So I just wanted to quickly acknowledge the people who did it. And this is my lab, and we get funding from the NIH and VA. So thanks a lot.
Thank you, Professor. Merci, please.
Well, thank you, Thomas, for a very good talk. I have two questions. The precursor cell for osteoblasts, the mesenchymal stem cell, does it derive from the bone itself, like the bone marrow, is from the adipose tissue surrounding it? That's the first question. The second one, about the citrate metabolism. Could it be a futile cycle, glucose in, citrate out, so to say? And can you speculate on that?
I'm sorry, the mesenchymal cell in bone marrow, did you say, does it exist?
No. Where does it originate from? Is it a bone marrow derived, or does it come from the surrounding adipose tissue?
No, it comes, it's still controversial, but most data would suggest that whatever the true, there's probably no true stem cell, it's probably a number of stem cells, and they derive from different places. There's going to be probably the adult, if you look at the adult bone, there still has to be some type of precursor cell that can, get out of its niche and start dividing because you need to repair fracture. Those cells do reside in marrow close to usually a blood vessel in a niche. The ones that are formed and act during development come from either the growth plate itself, believe it or not. There are papers out there that suggest that some chondrocytes directly transition to osteoblasts, and others probably reside early on the surface, the perichondrial surface, as that primary mineralization, as you heard from the long bone talk, as soon as primary mineralization starts, there's some precursor cells there that become osteoblasts, and they mineralize that site. So there are a number of different locations, and in adult it's different from the developing bone. And then the citrate, I'm sorry, I didn't hear you.
I mean, could it be so that it's a futile cycle, that you have glucose in and citrate out, like in adipose tissue, you have pyrite in and lactate in and pyrite out the other way, and so it's like a futile cycle?
So it looks like what's happening is that osteoblasts are these types of cells that use aerobic glycolysis preferentially, so they need a lot of glucose coming in, but they convert it to lactate. And so that allows the TCA cycle, this is theory now, it's not proved, we're trying to prove it, that allows the TCA cycle to keep making citrate at the expense, so it doesn't have to generate the ATP from the citric acid cycle, it'll go through glycolysis and then right to glycolytic pathways.
Any question? Yes, please.
Dr. Clemens, thank you. So are we still talking about balance between osteoclast and osteoblast for formation of the bone? And what you are doing is actually looking into the factors which make this balance, maintained or not maintained for a normal clinician. Listen to your lectures. We always thought about bone, osteoclast, osteoblast. We don't know much about osteocytes, but always we've been trying, the way we explained for the patient, that balance between osteoclast and osteoblast is actually what makes your bone good or not good. So you have mentioned a few of the factors which affect that balance. So my question, if this is what you were talking about first, and then my other question is, you said one of the problems our bones weaken after 20 to 30 years of age, that the osteoclasts make big gap and osteoblasts are not able to do that. Why this is happening in a normal, person?
Let me take that one first, because we're not supposed to live this long.
Wow.
It's clear now that, really, that's part of the problem. I mean, we lose bone. Now, again, what really you're asking is what confers peak bone mass, because that's really the important part of it. Because people can start out with a higher peak bone mass and lose bone throughout their life and be 80 with no problems at all, but other people starting out with less will get to that fracture threshold very quickly. And so I think we talked about genetics versus nutrition versus, environmental factors and so forth. I would suggest that the genetics is really strong. Looking at the GWAS data, it's incredibly strong that that's really the main predictor. By the way, muscle really does control directly bone mass. There's really good instances where muscle and bone mass go together. It's not clear whether muscle, just the contraction of muscle creates a mechanical effect which works on bone cells, or whether there's factors, but that's the other thing. Now, in terms of osteoclasts and osteoblasts, you're absolutely right. That's still, that's proven to be really true, and that's why I started off by saying kind of where we stand with that, understanding how that works. And really, the drug discovery process has been has really been successful because we knew a lot about that. And so as you up until, as you in your adolescent period, and in a mouse in the first 6 weeks, you don't have much remodeling going on. The osteoclast level is low. You have osteoblasts working, and they model what's called model bone. And then after that time, it shifts a little bit, and then the osteoclasts take over. And remember, you're trying to do good things. You're trying to repair damage. You're trying to release calcium when you need it because your parathyroid, you have low calcium, and your parathyroid gland goes up, and you have to mobilize calcium. But then I really don't want to be coy about that, but I think we are living so much longer than people did, back when these processes evolved in humans, that we never gotten these problems. And that's what we're having now, but that doesn't mean we can't cure them.
Nobody have a question? Yes, please.
Well, women, as you said, are more prone to lose bone mass as men. Is that due to the hormones or some other sort of imbalance between osteoblasts and osteoclasts? Why are women worse off in this case?
Yeah, there's still people that argue about this, but my own view, and I think it's pretty well held up, most people, most bone biologists believe that the main difference, if you think about those curves that would parallel down as you age, over the menopause, as women go through menopause, they lose the action of estrogen. Okay, estrogen, you lose your ovarian function, estrogen goes down. Estrogen is an extremely potent inhibitor of the osteoclast. Still not quite clear how it does it. But if you lose that inhibition, then osteoclasts become more active. And so the lines would diverge, and women would go down now to a new separate line, and therefore be at fracture threshold quicker. That seems to be the difference. But loss of testosterone and androgen action in men produces the same type of thing. In other words, it shifts the balance from the osteoblast to the osteoclast.
Yes, please.
Thank you. Where citrate has the role in this process?
Repeat, I'm sorry.
Say we have said about osteoclast, and citrate. Where the role of the citrate? Citrate.
Where does it made?
The role of citrate in the bone.
So the view is, and again, it doesn't come only from our own work, but citrate has been shown to be an important component of the crystal, the bone crystal. And it's still not clear. We're trying to prove it now, but it's been found 80% of all of your body citrate is in your bone. And the NMR studies have shown so far by modeling that it's too big. The molecule of citrate is too large to go in directly into the crystal structure. Rather, what it does is it caps the crystal growth, such that what you have is these leaflets of hydroxyapatite, which, by the way, give bone its strength. Collagen gives flexibility. Mineral gives it strength. Those leaflets then are too thick or too thin, depending on the citrate content, because they're not capped. And that disturbs the crystal structure and therefore the strength of bone. I'm sorry I didn't explain that, but that presumably is how citrate stabilizes bone structure, and we believe it's an osteoblast function to put that citrate in.
Thank you so much for the presentation. I would like to ask how we can translate the skeletal bioenergetic in terms of therapeutic messages to health professionals when it comes to the prevention from diabetes or management, the management of diabetes.
Good question. And I'm not a diabetologist and didn't think much about diabetic bone disease until I started to work on this project, but it actually is a good thought. First of all, let me say about humans, you don't see, and not that people they didn't look very carefully, but you don't see disastrous effects on bone with nutritional deprivation until you get to anorexia or when you disturb hypothalamic pathways, for example, in marathon runners, then you see low bone. So you have to have pretty severe malnutrition to get real devastating effects on bone in the normal person. Now, in diabetes, it's a real problem. I don't know the people who study diabetes in adults, they're in type 2 especially. Bone mineral is normal in terms of mass, but but its quality is extremely poor. It fractures very easily. And there's lots of reasons why that could occur. We don't understand it, but one of the things I'm intrigued about is could it be, by the way, the TCA cycle pathway I showed you with citrate is insulin dependent. So wouldn't it be interesting if part of the diabetic bone disease problem related to lack of citrate in bone? It's going to be something like that, but it's going to be more complicated. So in the terms of the normal take-home message from my talk, it's too early to say how each of these individual pathways I talked about regulate bone and body composition in normal humans. Like I said, we need more data. But in severe cases, it's clearly there. You malnourish somebody, they get bad bone. And then with respect to diabetic bone disease, it is there. It's a real problem. People don't, it gets a second-rate classification. People don't talk much about diabetic bone disease because there's lots of other problems. But it's a real problem for diabetics. The incidence of fracture is very high, and again, it comes from poor quality, not quantity.
Maybe in this case, we need to emphasize about the role of nutrition earlier and before we reach the peak for the development of our skeletal muscle.
Yeah, I should have mentioned that. I mean, I should have mentioned nutrition is really important. Now, again, we've thought of nutrition, we've known about that has been studied for a long time. Vitamin D, I used to work in that area, is a very good example where if somebody doesn't have enough vitamin D, what does vitamin D do? Well, it really has been proposed to do a lot of things, but really what it does is get calcium in the blood. So you need it for mineralization, otherwise you get rickets. But there's other nutrients that you need, calcium being one. And if you're really undernourished on nutrients, and not just total fuel, but certain nutrients, you're going to have bone problems for sure. So yeah, and building bone up to 20, you have a genetic component, which I think is large. You have a nutritional component, which again, only really if you're under-nutrition, you have something you're missing, you're going to have a problem. And then there's other things, like you can have other endocrine problems or diseases where you can have environmental toxins and so forth, but those are minor. It's going to be genetics, nutrition, to build bone into peak. Because if you could find a way to increase peak bone mass in everybody, then this issue of aging in bone would go away because you could still lose bone over a certain course, but you wouldn't fracture. So I think the emphasis should be on making sure the nutrition and the endocrine problems are not there. In other words, they're adequate. They have adequate diets, adequate nutrition, adequate amounts of vitamin D to build bone.
You have a question?
I go to from Munich. What's the role of bone density on metabolic syndrome? So on the time axis, is there any predictive value as well then? So we're talking about if you have diabetes, you have a high bone fracture risk, but if you're going first bone density and then metabolic syndromes?
I should know this better. I know that type 1 diabetics too have something wrong with their bones because they heal fractures very poorly. But it's complicated. And in type 2, of course, it's more complicated because you have these secondary events that occur in association after you get resistance to insulin and so forth and changes in blood glucose. But what is clear is that when and I don't know when it would start, and I'm not sure when you have what classification of diabetes type 2 you would have to have, but I know in severe type 2 diabetes, there's a point at which there's a tremendous weakness of bone, and it's not due to mass. There are studies out there that show that there's lower bone mass in diabetics, but they're very small. Most of the good data suggests now that bone mass in type 2 diabetics is normal or even sometimes elevated, but they still fracture, and it's the quality of the bone matrix and its mineral that seems to be compromised in diabetics, and that leads to a higher incidence of fracture in type 2 diabetes. I don't know, metabolic syndrome, I'm not sure if they've distinguished between different types of type 2 diabetes.
Any question? Yes.
Any other questions for this speaker? And thank you very much for this afternoon's lecture. Really very excellent.